Hi, this is Becky Carey taking over for Tina. In these next videos, we'll be looking at how to use Genome Browser. As Tina said, we'll start by clicking the Genome Browser link from the BLAST results page, and we'll end up on a screen that looks something like this. At the top of the page, under the reference sequence heading, there are 8 lines or tracks. The 2 multicolored lines in the middle are the 2 strands of DNA, with the colors indicating the nucleotide sequence. If we zoomed further in, it would show the corresponding letters. By convention, the top strand is shown with its 5' end on the left and its 3' end on the right. This strand is called the reference strand or the Watson strand and will be explained more in lab. The bottom strand has its 5' end on the right and its 3' end on the left and is referred to as the Crick strand. The 3 gray tracks above the DNA show the amino acid sequence you would get if you translated an mRNA produced using the top strand as the coding or non-template strand. There are 3 tracks because there are 3 possible reading frames. If you haven't gotten to translation yet in lecture, this will make more sense later. Again, if we zoomed in more, we could see the actual amino acid sequence. What we're seeing at this point are just the potential start codons in green and stop codons in red. The 3 gray lines below the DNA are the amino acid sequences you'd get if the bottom strand were the coding or non-template strand. We'll look at this more in the next video. Below the reference sequence information, we have the All Annotated Sequence Features track. This is the main one we'll be using. Below that are a few more tracks that we're not really interested in, so we'll get rid of those by just clicking on the X. If you accidentally close out the All Annotated Sequence Features track, you can get it back by going up here to Select Tracks, then typing All Annotated in the search bar and pressing Enter. That finds it for you, and then you can check the box and click Back to Browser. Now that we've gotten rid of the extra tracks, the first thing we're going to do is highlight the region we're currently looking at. That way we can scroll or zoom and still find the place we started from. So to do that, we'll click View, Set Highlight, and it defaults to the current view, so we just click Highlight. If we zoom out a little, the yellow region in the middle is what we were initially looking at. The reason that it's important to be able to keep track of it is that that region is the part of the yeast genome that matches the sequence from our SNAP-gene file. In other words, this is the piece of the yeast genome that we managed to sequence, and it's one end of the insert in that particular recombinant plasmid. To see that a little more clearly, we're going to add a track that will show us EcoR1 sites. So to do that, we go to Track, click Add Sequence Search Track, type in the EcoR1 recognition sequence, GAATTC, and click Search. So now we can see the EcoR1 site at this end of this particular fragment of the yeast genome. The reason it shows arrows going in both directions is that it's a palindromic sequence, so it's found on both strands. To get its coordinates, which should match the ones from your BLAST result, we can right-click it and choose View Details. This data window shows us that it starts at nucleotide 46287. If we look back at the BLAST result, we see the same coordinate. We'd need this information to, for example, determine the precise size of this yeast genome fragment. We're also interested in looking at BAMH1 sites, so again, we'll go to Track, Add Sequence Search Track, type in the BAMH1 recognition sequence, GGATCC, and click Search. Looks like we don't have any BAMH1 sites in this region. Most of what we're going to be looking at will be in the All Annotated Sequence Features track, which we'll explore more in the next video.